Okay, so we'll move to Maria.
Yes, I'm Maria Jose Diez from UPV also. I think everything has been explained by my colleague Jaime. I work with him in Solanacebus crops mainly, and we lead the work package 8 in this project, and we participate in other work packages.
Okay, after Tony.
Yeah, I'm the coordinator of the project. I will be mostly working on the work package that people don't like. I mean, but the coordinators and ethics, but also in the work package 7, the quality traits. And I guess that my participation here because previously I coordinated Traditom, and we are going to transfer lots of the information from Traditom into work package 3.
Okay. And Tony is also here to see that everything is going smoothly as coordinator. Okay.
I'm sure.
So Giovanni.
And especially in an ethical way.
Yeah. Okay. So Giovanni, so you are in the order.
So I'm Giovanni Giuliano. I work at the ENEA, and I coordinate work package 4. And the day-to-day management of our interactions with work package 3 will be done by Giuseppe, who is our bioinformatician.
Okay.
So Giuseppe, you want to introduce yourself?
Yes, Giuseppe.
Okay. I'm Giuseppe Aprea from ENEA, Italy. I work with Giovanni, and yeah, will participate to work package 3 activities.
Okay. Thank you, everybody. So we will start our meeting. So the goal of this meeting was to start to work in the frame of work package 3. So we had some meeting with some of you, and I heard also that you had in the other work packages, you had some meeting and you talk about the data and how to feed work package 3 with data. Okay. So the goal of this meeting is to discuss between us how to manage all this. So we have happy to have Phenom here with us to remember you that this work package is co-led by Toulouse and by we say by Rehovot in Israel. Okay.
Rehovot, yes.
Rehovot, okay. And we will work together to make the best tomato database in the world. So this is the ambition. Okay. And to have an idea. So the first thing maybe is to discuss which kind of data we start to work on, because we have here coordinator of the other work with the other projects, EU projects. We have so Traditom, we have G2PSOL, mainly our TomGen, because our lab is coordinated TomGen. We have also Brisolve. Okay, so from all these projects, there's lots of data that are generated. So the idea is to start by something. So to start by the data that come from this project, and so there is two things. So is to decide which data we start to store and to organize and to link to a web server to watch and everything. And also, because the other work packages in HARNESSTOM will produce data also in the following years. So the idea also is to discuss with us with the format that we have to deliver this data to be compatible with the platform. Okay. So these two main things. So from for discussion that we had with some of you, we discussed mainly with Clara that have a good experience with Traditom. So she was in charge for doing the database of Traditom. So she knows very well also the data that come from this from Traditom. We have also so people from Yaniv team that also work on G2PSO that have also a lot of data. So now the idea is to be aligned about the information we have in the discussion that you have in all the other work packages. Maybe I will I will let maybe work package leaders from the other that to give us a summary of what you discussed and what you expect to bring as data, and also the leader of the other EU project. So just to tell us what is their feeling, which data we will start to introduce in HARNESSTOM. Okay.
So maybe we can start with Work Package 4, if you agree.
Okay.
Right now, Work Package 4 has not generated any data, but there are data that have been already generated from G2P Sol. They are essentially data for genotyping of about 15,000 worldwide accessions of tomato from different gene banks, including all the passport data on these accessions, etc., etc. And these data, to my knowledge, are already on the gateway, right, Amnon?
Yeah, trying to unmute. Could you repeat the question?
So the data from the low density genotyping from G2P Sol, are they already on the gateway, or are they being uploaded right now?
Currently, from the core collection, it's all there. Of the core collection, it's all there, and the rest is not yet there.
Okay, so but it's being uploaded.
Yes, it's in the process of being uploaded.
So
yes.
This is a big chunk of data that is already ready from a previous project, and in the coming months there will be 10x sequence data or 15x sequence data of the G2P Sol core collection, which is composed of about 400 accessions, and this will be uploaded on Phenom, so it will be automatically transferred, hopefully seamlessly transferred to HARNESSTOM. And the phenotyping data of the core collection in different locations have already been uploaded or are being uploaded again on the G2P Sol gateway. So this will be also transferred. So this is a big amount of data. So what we expect to produce in HARNESSTOM Work Package 4? We expect to produce more SPET data. We expect to produce very low density barcoding SNP data based on amplicon sequencing. And we expect to produce very high density SPET data, like 100,000 or more SNPs, all in the form of VCF files, etc., etc. So pretty standard format. And additionally, from Work Package 4, I expect also some phenotyping data to come from the activities on the recombination frequency and the speeding of the regeneration time, but this will not be major. And of course, all this will comprise also mapping of the traits, both from the G2P Sol data and of the data generated in Work Package 4. So chromosomal location of traits. So that's more or less the data we already have and will produce.
Okay, so let me welcome Yaniv just to join us.
Hello, everybody. Sorry,
okay.
I'm late.
So maybe Yaniv, you can present yourself to the team, please.
Yes, sure. So my name is Yaniv Semel from Phenom Networks in Israel, and I work together with Amnon. Here is also with us, and we are responsible on managing the database and loading and analyzing data and so on. So nice to meet you.
Okay, good. Okay. So is there any question for Giovanni? I have some, okay, Clara, we will start with Clara.
Yes, regarding to Giovanni, and I think that we have in Traditom and in other projects, is we have genotyping data by GBS with SMPs. So which is the format that we need to upload the data in the database? A HapMap, a matrix with HapMap data, with FCV data, which is the format that we need to upload all this data. I think that this is the most important, no? That we need to know. Yes. In this case, Giovanni had talked about genotyping data, about passport data, and maps. So yes, we need to know the format to upload all this data.
Yeah. Okay.
So what kind of matrix? And and in the case of passport, I think that GTP have also the multi-crop passport descriptors because it's Maria Jose there. So I think that you are using this like us in Traditom. So I think that we need to define for this that is common to most of projects, the files that we need.
Okay, good.
Well,
so I think this.
I think the best suited person to answer this question is Amnon or Yaniv.
Okay. This is what I want to say, maybe because you already have the data organized with the huge data from G2P. So maybe you have to use this format to include the other data.
Yeah, so we can send an example of the format of the data that is needed to load into the system. So we can send it separately if you need.
Okay.
Alternatively, I mean, if you have the data in any format, you can send it to us, and if needed, we will reformat it and load it. Because in the G2P Sol, I mean, the way it works is that eventually we receive the Excel files and we manage the upload. It was not done by all the other members. And I don't know how we want to work here, but anyhow, either case, either you can send us the whatever format you have and we will manage it and load it, or alternatively, we can send you the required format to be loaded. So both ways are possible.
Okay. Just a question for Giovanni. So you told that that we have 400 core collection with genotyping. It was GBS that you did in UT.
It was always SPET.
SPET, okay.
It's SPET, low density SPET. You probably know what the technology is.
Yeah. You published it in a paper. Okay.
And it's a targeted technology that also sequences the region around your target SNP. And so it allows flexibility. If you use just the target SNPs, you get, for instance, if your target SNPs have been selected to be intra-specific, you explode the diversity of the Lycopersicum clade. If you use all the SNPs that you discover, you compress Lycopersicum, but you get a more clear picture of the systematics of the whole subfamily of the Lycopersicum of the wild species. I should add that very small scale, we did also some SPET genotyping of couple of hundred Traditom samples. So for these couple of hundred, we have both SPET and GBS data, of course, generated by Traditom. And now we are in the process of doing a few tens of TomGem samples.
Okay.
So these will be also included. So it will be mostly G2P sold, but also a few hundred from other projects.
Okay, this is good. And for the phenotyping of the core collection, so which kind of phenotyping we have?
So maybe Maria Jose and Jaime can explain better than me, but the descriptors were largely were largely taken from the Traditum descriptors, so there will be lots of coherence between the Traditum data and the G2P Sol data. On top of that, there will be metabolic profiling data that will be generated by ENEA physique on a voluntary basis. They volunteer to do volatiles, and Max Planck.
I just have to say that they will be jointly by UPV and CSIC, because they will help us to do it. Okay.
Yeah.
Okay. Okay. Good. So good. So maybe we can.
For the metabolic profiling data, since the partners involved are the same as the ones in Traditum, probably the whole system of presenting the data will be homogeneous between the two projects.
Very good. Very good. So now we have a nice idea about what has been done in G2P Sol in terms of resources and data, and what it will also come in HARNESSTOM with other kind of phenotyping like metabolomics and so on. So maybe we can move to Tony or Clara. Yeah.
I think I was going to tell you about not moving to the next work package, staying in the work package 4 and talk about what the contribution is from the other projects.
Yes.
So Clara maybe can summarize what type of data we have from Traditum that we can transfer to HARNESSTOM.
Yes.
No,
in Traditum, we have GBS about 18,000. My numbers with the thousands. 1800 accessions, and we have also phenotyping data for about 15,000. And what else? Phenotyping data for descriptors, and phenotyping data for tomato analyzer, and also for a core collection we have descriptors also for yield, earliness, and some physiological disorders. And all of this is structured in different files. One is for experiments with all the conditions that were done in the field. The other is for the type of traits and the scale and all the description for the files, and then the phenotyping data and the genotyping data. We have also data for metabolomics in 4 platforms. This is done only in the core collection. And I don't remember what else, but we have all the data organized, as I told you the other day. So it's really to send. Also, we have curated data and inputted data for because we are analyzing the data. So for some of the traits or some of the matrix of GBS matrix, we have data inputted. So we have raw data and curated data.
Very good. Okay. Good. Yeah. We have a summary. I will after maybe make a folder in our platform just to share all documents that we will receive and summaries from this data that come from each partner. Okay. You know that Clara sent us a PPT that is nicely described, and also Jaime also. So I will put this in a folder so we can share it with all of us. Okay. You have question to Clara? Okay.
Yeah, Simo. We were talking the other day about how open this data can be to the partners, of course, open.
Yes.
Rest of the world to maybe flag the data in different colors, red, GLE, or green, whether some of the data we want to keep still private within the consortium because it has not yet been published. Okay.
Tony, okay. So I wanted to discuss this point later when we'll have all an idea about the data, and after we'll see the strategy. It's a question of strategy and also techniques, how we will work how to store, which place, if we have to have two platforms or one in one place, and we will work all together.
Yes.
This is strategy. We will maybe discuss about it later.
Clara, okay. Was also something we missed is that we have been working on the data and produced GWAS data also associated with combining the genotyping and the phenotyping data. This is already underway, and we already have most of the results already processed.
Okay. Okay. So, anybody have a question to Clara, or is it clear?
Well, I would like to make a comment also. We are forgetting to comment about pictures. There are thousands and thousands of pictures that also provide a lot of information.
I forgot it.
Yeah, and is additional to the picture is all the scans from the tomato analyzer also.
Yeah, but it's including the pictures.
Yes. Yes. There's the process information from the tomato analyzer and the raw information. That is probably the largest one.
We had this discussion with Clara last time. If we have to store only the scheme of the fruit or the whole picture, so we will sure we have to store picture because it gives a very good idea about the phenotype. Okay, but they are of big size, so we need also to do it in a good way to make it smoothly when we want to search the data. Okay. So maybe we can move to PV, Jaime or Maya. Okay.
Yes. For the project Brezov, in which we are participating, of course, the amount of data is much less because it's not a tomato project, it's a vegetables project, of which tomato is one. But basically what we have is phenotyping data for 325 accessions that have been phenotyped with a phenotyping kit, which is very similar to the one from Traditom and G2P-Sol. In fact, in the collection that we have in this project, there are 12 accessions that are shared with both Traditom and G2P-Sol, and other 13 with Traditom and 18 with G2P-Sol. We also have data, or will have 42 accessions that have been grown in different environments, controlled drought and low nitrogen. And for this, we have phenotyping data, but also we will have metabolic ones. Also, we have data for tolerance to Fusarium and tomato spotted wild virus for almost 200 accessions. And the data are stored in Excel files. We don't have database yet in the project.
Well, this is a good opportunity to put it with the other data. Okay, it's very good.
Jaime, could you repeat the name of this project? I didn't catch the name of the project.
Brezov, B-R-E-S-O-V.
Thank you. I'm just trying to notes. Thank you.
Okay. So we maybe move to Mathilde, to Avignon. So it's good because we are traveling with Zoom. so we can see that it's a sunny day in Avignon, but I think your microphone is muted. Okay.
I'm sorry. It's nice to travel like this, but it would be better to meet in one place, I was saying. Yes. So for work package 7, we have some data which were previously published on GWAS and QTLs for food quality traits, mainly volatiles or primary metabolites. And well, the data are usually provided either in as supplemental data in the publications or in dataverse system from INRAE. But so they are already organized, I would say. And we have some unpublished data of GWAS for volatiles, which are interesting us for HARNESSTOM. So we have many GWAS results. We have also the meta-analysis that we published last year, gathering the data from the publication of Timan and 2 publications from our group, and all these data are available. What else? We have some data also which can be helpful for work package. The environment, it is 5, or is it 6? I don't remember. 6. Thank you. So we have some data about the magic populations that was assessed actually in 12 environmental conditions, either control or heat stress, water stress, salt stress, and all these data were published. So it is a population of 300 accession, magic accessions, and which were genotypes with 1300 SNP markers. And for the GWAS, most of our studies were genotyped with a SolCap array. So it is about 7,000 SNP. And we have some old QTL data from recombinant in breadline population, which was genotyped with 500 SNP markers. I think that's it, more or less. I have a...
It's a lot of data, it's good.
Yes, it's a lot of data, and maybe one of my main question is, well, I think that for genotypes, it's more or less easy. We have 0, 1 or AGTC, but it's not so complicated. Maybe the most complicated will be with QTL, where we need to discuss together which format do we want to put and which data we want to put. And we already sent you some ideas that we discussed with Tony Monforte. And I think because we will need it very, very soon, also this data, and to be able to manage the first crosses and to check which crosses we will do, etc.
Very good. So it will make a link with that. I see that Tony Monforte is shaking his head. I think he want to add something about
this. Okay. So just that we were discussing how to compile the QTL data, because I mean we have also some idea. So I think we need all of us the same template for the QTL, so we can start to fill out, because we have a lot of QTLs. Yeah, lots.
Yeah, and also you point a good question last time is about to have a tool in the database that can move from one reference genome to another one, because maybe this QTL are on one reference genome. We are now in 4.0, maybe we'll have 5.0 after. So yeah, this we have to. Yeah.
Tony, did you see the templates that I sent you yesterday for the MICAS? That is one of the standard templates for QTLs and associated analysis for the EBI, and the other is for the GEMPs from the INRA. That they have standards, they are doing the standards for QTLs. They include also for experiment data and population data, maps, markers.
We took some ideas, but I think that it's better that the template is just one sheet.
Yeah,
because several sheets.
No, just to give you ideas.
We got some. I mean, we prepared yesterday a number of traits or things that give information that but maybe included. I will distribute this template with you, so you may think we can add something or remove something, because I mean we have a lot of info for each QTL. We have to add a lot of information, like population, experimental design, the traits. So we need to have a clear trait ontology, so we can share the trait. So same ontology for the same trait to compare. Also, I think it's important to have an experimental ontology. So what kind of experiments? Experiments, greenhouse experiment, we need this ontology for stress conditions. What kind of stress? So
Yeah.
But Tony, there are different databases with trait ontologies and experimental ontologies. So we need to go to this because
Yes.
they are they
Yeah.
have codes and
exactly, that's what we have to. I didn't go to these databases, but it's something that we have to have very clear.
Yes. So HARNESSTOM is the opportunity to unify all this data that are in the different places. Yeah.
But in some cases, it will be also difficult to unify things, as we will not reanalyze everything, etc. So maybe we have also to discuss what is mandatory and that has to be unified and what is more free. Because we will not be able maybe to describe everything as expected.
I agree. Yeah.
I can also share, but I don't know if it is the files that you shared, Clara, the files that we used in GNPIS in INRAE for GWAS data. It is an Excel sheet where you have all the metadata, the genotypes, the kinship, the structure, and all.
Yes, you have different sheets to fill.
Yes.
I think that is not necessary to fill all,
I
but
can
some...
share this sheet if you want. So you
I
can.
have this. I have this, and I send also to Tony. And I have also the links to go to phenotyping data, to all the types of data you can upload.
Yeah.
Okay. And it's easy because I think that these sheets also, you fill one
field, and we already spent hours discussing about this sheet in INRAE when we produced it. So maybe we don't need to spend again some hours to discuss about it.
Okay. So yeah. So also we thought that maybe it's a good we can work this way, and also Tony Garnelli has suggested this also, is to have what we call in other projects captains of some kind of data. So somebody from our group that is aware about the problem and he can it's as an expert, let's say, that can with whom we can work. For example, for QTL, maybe we can choose one that can be somebody who can think more and maybe organize this kind of activity. Okay, do you agree for this?
Simo, in each of the work package 5, 6, and 7, there will be people reviewing the information that is already published, or in the case of each group, the literature or not. So there will be already, let's say, people already involved in this QTL, in these work packages. I don't know if it's we need to have one that serves a connection with the work package 3, or each work package already has the people who will, let's say, working on.
Yeah, but what we need is a template to fill out to have a template so we can say to the other partner, okay, you need to fill out this template. Let me if you miss anything. But we need something to start. So maybe I think this maybe we can have a dedicated meeting for this question. I think. Yeah, I think this meeting is more general. Yeah.
Okay, so also to give you an idea about the data that we have in the lab, okay, because so we are just to have a general idea about the data that we can put in the platform. So as you know, so we have the data that come in from the phenotyping of the accessions of TomGen, okay. So in TomGen there were many phenotypes in many places. For the, I think it's 700 accessions. So we have some phenotypes of those. So they're mainly about yield in experiments that have been driven in Italy, Spain, and other places. Okay, and also we have an RNA-seq database, a big one that we call TomExpress. So inside there is a lot of RNA-seq data coming from many other projects, not only TomGem, but also other published data. So just to give a summary about this for people that don't know it.
Fassimo, the RNA-seq data is published data. So the quality control is that is published.
Yeah, and we have also data that is not yet published also inside this platform. A publication in a journal is not mandatory because we have some collaborations. So, for example, Mathilde, she sent us some data that we put them in Tom Express, but now we will have the paper that will appear. So this data will be public. But the RNA-seq data are already in the database, and they are flagged as not public, but they are already inside. Okay.
Excuse me, I have another thing coming up.
Okay.
I will leave, but Giuseppe will stay with you until 10. If there are any issues.
Okay.
Have a nice meeting.
Bye-bye.
For the future, if you could make a doodle before fixing the time, because everybody is
busy. So this is why we try to do our best to have a very quick meeting, at least with the people that
with these teleconferences, every moment of our life is is
you know Eden, she prepared the doodle, but after we were thinking I think it was good to have one first, just to start, and after of course we will organize things.
Maybe before you leave, Giovanni, I wanted to throw out this idea I have that maybe and probably you will like this. You have like kind of a hackathon kind of meeting with the people who have been working with databases and so in the projects that we participated on or not, like UTP-SOL, TomGem, Traditom, and so on, and also maybe inviting the people who will be doing this for other projects in the same call that were granted at the same time as Traditom. Maybe would be a good idea to have 1 or 2 days hackathon
as HARNESSTOM.
Yes, at the same time, but also the people who already did similar job in previous projects.
Yeah, I mean, there is certainly, I can think of other projects as well, like the project on cereal and legume genetic resources.
Yeah. These are having granted about the same time or before the call before, right?
Yeah. The main challenge there, in my opinion, is finding the right size, because if you make it too large, it will end up in a mess. Everybody has her own idea.
Yes.
So the main challenge is to find a group of people that, many of the people are already in the project, but you can cherry pick a couple of capable people outside the project that can give some.
Yes. Okay. We will do a kind of selection before we contact them.
Yeah.
We already are in contact with the group from Italy that is coordinating a berry project, another one that is doing cereals and legumes. So let's talk there. Okay, but I think it's a good idea that many other people will be trying to do a similar type of databases for other crops, that maybe we can to meet them and see how they are going to do it. But basically the ones that are going to develop the database.
Is Richard in this project?
Not that I know.
Well, he would be an obvious candidate.
Yes, of course. Yes. Okay. Thank you, Giovanni. Bye.
Bye-bye. Okay. So I think now we have a good idea about which data we have in each places. Okay. And maybe now we can talk how we will organize ourselves to make the platform. So in the project, it was decided to make a gateway, a public gateway that gather this data. So I just made a summary. So we have already a database in INRA, okay, that have already data that coming from Mathilde's lab. Is this right, Mathilde?
Yes, but as people from GNP-IS are not partner in this project, I don't think it is good to try to use this.
Okay.
And I just collaborate with them at the beginning of their GWAS database, and then they focused more on serials, and we stopped working with them. So we just have the templates, but that's it.
Good. Okay. So, and also we have so the big database of Phenom. Okay. And now
Simo, going back to there was this idea that maybe what is the capability, the storage of this database, because you were telling that maybe the tomato analyzer raw data or any other raw data that is based on pictures or images, maybe not good to, or maybe they can be stored but not been, let's say, analyzed during the processing of analysis.
So this is a particular case, but we have other data that we can store, like the genome sequences and the markers and the QTLs and all this kind of, or the VCF files of genotyping. So I think this will not make problem of space. Okay, not big problem. Yeah, the picture is a particular case. We have to think about it. And so Tony, when we were discussing, you mentioned this idea to flag the data that can be public, data that maybe, can be, as a machine to use colors, green, we can publish, yellow, maybe not, and red, not to make it not public, okay? And also, so we have to think if we have to make one database, okay, that is as we said in the project, that can be at Toulouse in our institute. We have the capacity for that, okay, and to put all this data that Giovanni said and all of you said, and to connect them to be able to do all the analysis that we listed in the data in the project, like visualization of the QTLs, visualization of genetic map, visualization of phenotype. Okay, so now, so what is the strategy? Is to put all these also the data that are in a G2PSOL database in HARNESSTOM data gateway. So how you see this, Yaniv?
Yes, I would try to get the data from the other sources and add them into our gateway. Yeah, I would definitely go for it.
Okay, so because you have already data that are stored and you use connectors like BRAPI, and so we'll discuss this maybe in more details. We will have a separate meeting, technical meeting.
Yes.
So we can, in our side, make also connectors and work together, you and us, to develop the HARNESSTOM gateway to visualize and to analyze all this. What Giovanni said just before, the data that come from G2PSOL and also from the other project. Okay, good.
Yes.
I think it's good that the whole set of data that we put together in HARNESSTOM coming from other projects have a mirror or have this exact replicate in Phenom network. I think the idea is that we have a public and a private partner that have the same data as we produce in HARNESSTOM or analyze in HARNESSTOM. Because we don't know what's going to happen with Phenom networks. Maybe it will be a 100-year-old company in 60 or 70 or 90 years. I will be happy for that, if I can see it, of course. But in the case of HARNESSTOM, we have the commitment with the EU to have a public database. also we need to have in mind that we discussed this in the kickoff, that it would be nice if this is somehow connected with the seed repositories, with the germplasm bank, so that whenever we have an accession that has been genotype, phenotype, or whatever else characterized in any of the projects that we are now putting all this information together in HARNESSTOM and in Phenome, that in this seed bank, they have this information in a click of their accession. In the other way around, that maybe when someone look at an accession in the Phenome network database or in the HARNESSTOM database, they can get access to where the accession is and who can provide it. So yes, interconnectability, I think, is important, that we, from the very beginning, at least we facilitate this. Whether this at the end has a cost for the seed banks to introduce these links or not, I don't know how easy this can be, but I think we should keep in mind that this could be the real value, to connect seed banks with information and information with seed banks. And of course, for me, the two databases in a public and a private storage will be the best thing, because I think many breeding companies would like to have the assistance and the help of Phenome to develop something, and it's nice that all the information is in Phenome, but maybe some public researchers prefer to have this information in a public database without going to the private.
Sure. Yes, we can work like this. I think it's the best way. Yeah,
I absolutely agree. I mean, I didn't mean that all the data will be stored only in Phenome networks. That's not what I meant. I mean,
no.
I just want to aggregate it and keep links also to other repositories, public database. Sure, absolutely, I agree.
This is a project. It's an innovation action. The best thing we can do is that the researchers benefit from it and companies benefit from it. Yeah, this is clear.
Yeah. Okay.
Okay, good, very nice. So somebody have a comment or question?
Yes, maybe to go to the next step, once we have all this data, we need to use them. And I don't know if we will use them with tools which are in Phenome, because I didn't go to Phenome since several years. But for instance, we plan to use two open source tools, which are Flapjack to manage the genotypes and the QTLs, to identify the accessions which carries the right QTLs, for instance, and prepare some back crosses or crosses. And also to use Biomarcator, which is a tool which has been developed in INRAE also to do some meta-analysis of QTLs, and which provide you with the genetic map and all the QTLs that you put in by chromosome, and you can link it to the genome of your choice. So it can be also useful. I don't know if these kind of tools are already available in Phenome, or if you plan to develop such things, or if we can just use those tools. I don't know.
Absolutely. That's absolutely okay to use other external tools, no problem. And I don't think that our purpose is to develop everything that is already available. That's not the purpose. Okay.
And particularly Flapjack, I used it in the past, but now it has been upgraded for choosing the crosses, and it is very nice.
Yes, absolutely. I've actually been looking into Flapjack, so we would like to integrate that into the website. Can you please repeat the second tool that you mentioned? I haven't seen that.
Biomarcator. I can send you the link if you want.
Yes, please. That'd be great. Thank you.
Yes.
I think
we are developing a new version which will be adapted to GWAS, because up to now it was just for QTL from segregating populations, but now they are adapting it to GWAS data and to put GWAS and together with QTL data, so it can be interesting.
I think it would be nice to have as many tools to analyze data as possible that facilitates the analysis of data on the site and so. But I also emphasize that the EU was, and this was part of the call, was very clear in the call that we need to implement visualization tools. This was very strongly indicated in the call. So maybe we can go without some of these QTL analysis tools or whatever, but that I prefer that we have them, but we cannot don't go without having some visualization tools implemented in the system. Okay, so implement.
Okay. Yeah. Okay.
Yes. So yes, as it was said, is that the idea is not to develop new tools, but there is many other many tools that are already available. So there is, when we discuss with Lucas and also Tony, so there is breed-based, for example, that is something that we can install also. I think there is one of your lab, Mathilde, who is in Lucas' lab that he developing this breed-based database. I met him in San Diego, and he is ready to help to install it. Okay. And yeah, there is many tools that we have to choose the best and the compliant one. So there is BRAPI also. There's some tools that are written in BRAPI model, let's say. So we will focus on those tools. Okay.
And it is maybe, to my opinion, the most important, because, for instance, I worked a lot with GNPIS in France.
Mm-hmm.
Actually, as they did not develop such tools, we feel that our data are just put in a database where we don't find them anymore. So I think it is really important not just to have a database, but also to have something to use all this data.
Yes. So this is the goal.
It is really necessary.
Okay. So Mathilde, we will contact them together to see how we can connect to this data or bring them in HARNESSTOM. Okay. Okay. We will do this. Good. So any other comments or questions? Okay, I think we have a good start now. So just to summarize, so we will so work together to prepare. As Tony Monforte said, we need to define templates and also to see which kind of data to start with, the minimum of data that can unify all of us, to start to download this data and to prepare them for the integration. Okay, and we will continue to work with Yaniv and Amnon to see how we parallelize it, how to mirror the data. If we maybe it's a way to do it is to connect to data in your place, just to make visualization for those who are public, of course, and that you agree that it can be used by in an open way, or maybe if there is problem, we can also imagine to copy them in our server, and if it makes things going quickly and smoothly, we can also maybe opt for this option. Okay, so we will try to work. We will work in a linked way together, and I think we will do something good. So we have a deliverable in month 6. Okay, this is it. Right? In March. So we have to write some kind of model of how we want to store this data. Okay, so we will. So we will work with Yaniv and Amnon, and of course Eden. Eden is 100% on the project in our lab, and we have also other people in our lab working on this project. And we will make a good model and propose it to you, and after we will continue to do our work. Yeah, as Tony said, we will focus first when we will have the model of the database. We'll focus on putting some data and integrating visualization tools, and in parallel we will also add the analysis tools. Okay, so this will come. Okay.
Yeah, one thing. Before we close the session, I think it would be nice to remember and put the emphasis that maybe we are going to have the same trait and the different names, or different names for the same trait, or we have different reference genomes. We are going to have different genotyping platforms. We are going to have different accession names for the same accession. We need to find out ways to navigate through the different names of the same accession. So each project has renamed the same accession and put the name of the project. Like in Traviton, we did TR accession. So we need to make sure that we don't get lost.
We must manage some synonyms.
Yes.
Yeah. I think Clara, she proposed to use the universal ID of accession. So I think there is a tool or website that propose this kind of unification.
This will be a new name for each accession.
Yeah.
So
another one.
It will not solve the problem of different, the same accession which has already different names. You will have different IDs. So
it has to be a tool that if you have, if you read in a journal LA 394, then you can identify whether we have something under this name. No matter whether in the phenotyping experiment we use Traditon names or UTP-0 names. So we need to be able to find an accession that is in our collection but maybe renamed.
Okay.
The other day I proposed you the permanent unique identifier. This is an initiative to unify and to have one identifier for accessions, and then you can put all the synonyms.
Yeah, this is the name that I was searching for, permanent unique identifier. Yeah, but yeah, we have, as Mati said, yeah, it will add another name, but we have to use, we have to agree to use one that can, on the top of which we will link all the other names. Okay? We need an ID that is unique. Okay.
I think the basic thing also, Simo, is that you have identified a contact person for each of the major projects that will be providing data to the HARNESSTOM database, and also a contact person from each of the work package that will be defining QTLs and so on from the literature, from the ARM labs.
Okay, so with Eden, we will prepare an Excel file that we will share with you with these names and to waiting for you to agree or to propose other things. Okay, so contact for each work package and contact also for if we don't have people from this work package but have data in other projects, we will also add this data. Yeah. Okay, good.
And as you said, it has to be the person who can provide the data and work on it. Okay.
Yes.
Don't come to me then.
Yeah.
Yeah. No joke.
People like Clara.
Yeah. Okay. Good.
Okay. Very good. So
we thank you very much.
We stop here. So I will send you after a link of this video if you want to record it another time, so we can remember what we said. And with Eden we will write a short summary by email to point the important things that we discussed. Okay.
Okay.
Good. Very good.
Thank you
so have a nice day. Bye bye.
Okay. Bye bye.
Thank you. Bye.